Next, we will discuss the steps of genetic engineering, Chapter 17 Biotechnology, Class 10 Biology. Which steps are involved in genetic engineering? The first step is isolation of gene of interest. Gene of interest means which gene you want to make the copies of, means the desired gene. Isolation means separation. Isolation of gene of interest is the first step of genetic engineering. The separation of desired gene or isolation of gene of interest is done by using DNA and special types of cutters, molecular scissors, special types of enzymes called restriction endonucleases are used for cutting a piece of DNA, means gene from DNA. A piece of DNA is called gene, and to cut this gene of interest from the whole chromosome, the special types of enzymes used are called restriction endonucleases. These are called restriction endonucleases because they are restricted to bound to cut just that gene which we need. Restriction endonucleases just separate or cut the desired gene. They do not do any cutting or harm to the other nearby genes. That is why their name is. Restriction endonucleases may also be termed as molecular scissors because they act like scissors, mean like a cutter. A piece of DNA or desired gene is isolated in the first step by using restriction endonucleases. In the second step, gene of interest is attached to or linked to a vector. The second step is the use of vector. What is vector? Vector in biology is any organism or a part of organism which takes one thing from one organism and gives it to another organism. A vector acts like a medium to take one thing from one organism and deliver that thing into another organism. And in genetic engineering, vector means an organism or a piece of organism, in particular plasmids or bacteriophages act as vectors. Plasmids are extra chromosomal DNA of bacteria. For example, this is a bacterium. A bacterium consists of two types of DNA. One DNA is called chromosomal DNA, and the other type of the DNA present in bacteria are extra chromosomal DNA. This is chromosomal DNA and these two are non-chromosomal DNA. These non-chromosomal DNA may also be called plasmids. The role of the chromosomal DNA is in bacteria reproduction, but the role of the plasmids in bacteria is controlling metabolism. These extra chromosomal DNA or bacteria are called plasmids, and plasmids are generally used for as a vector for taking the gene of interest from one organism and delivering it into another organism. Plasmids serve as a vector. The gene of interest is attached to a vector DNA, means a plasmid in most cases, by using some special types of enzymes which are called ligases. Ligases are joining enzymes, whereas restriction endonucleases are breaking enzymes. The piece of DNA which we have separated in the first step is linked into the vector DNA by using joining enzymes called ligases. At this step also endonucleases are used. In the second step, first we separate the vector DNA. Imagine this is vector DNA. We cut a piece of vector DNA. This very small piece of vector DNA is cut by the help of restriction endonucleases. Now this was our gene of interest which we have isolated in the first step. First we use this restriction endonuclease to cut a piece of DNA of vector. Then we paste the isolated gene into here by using joining enzymes called ligases. Now you see and you know the DNA piece from here to here is that of vector. It is vector DNA, and this very small piece is isolated gene of interest, because now vector DNA has a mix of two DNAs. Now vector DNA contain a foreign gene. That is why this vector DNA is now a mix, and it is now called recombinant DNA. Recombinant DNA is defined as a DNA which is a mix of two DNAs. When one organism's DNA is mixed with another organism's DNA, the new DNA is called recombinant DNA. This second step of genetic engineering is very much crucial and essential. Genetic engineering is also named recombinant DNA technology, depending upon the production of recombinant DNA, which is an essential step in genetic engineering. Genetic engineering is also called recombinant DNA technology because of this second step. Now, we will talk about the third step involved in genetic engineering. The third step involved in genetic engineering is insertion of recombinant DNA into host. When host, for example bacteria, have a recombinant DNA inserted or entered into them, they become genetically modified organism. This third step can also be called the production of GMO. GMO means genetically modified organism, an organism whose genetic makeup is changed because it is having an extra DNA which is not a part of its DNA, the recombinant DNA. For example, this was a host or bacterium before insertion or adding of recombinant DNA to it. So this is not a GMO bacterium. When we have added the recombinant DNA to it, which was like this, having a foreign gene, then this bacterium is now called GMO because it has a recombinant DNA inserted into it. This is not a GMO because it is not having recombinant DNA in it. But this bacterium or host is recombinant DNA, and because of the presence of the recombinant DNA, it is called genetically modified organism or GMO. Now comes the fourth step in genetic engineering. This fourth step of genetic engineering is called growth of GMO. Growth of genetically modified organism is made possible in this step by providing the genetically modified organisms proper growth medium and proper environment for much and more growth of genetically modified organism. During growth of GMO, this fourth step, a large number of genetically modified organism formed. From this one GMO, there will be formed two genetically modified organism, and from 2 there will be formed 4, from 4 to 8, and so on, in a very less time, because an optimum environment is provided. A very much large number of bacteria or genetically modified organisms are produced. In the fifth step, which is called expression of gene of interest or the expression of gene in GMO, This is the last step of genetic engineering. In this step, the gene which is present in GMO and which has been taken from outside the gene of interest in the first step, the gene we took, expresses itself, and the plasmids or vectors or the host or the bacteria or GMO which we have used for this purpose doesn't know that it is actually replicating a foreign DNA. It takes the foreign DNA as it is the DNA of the host. That is why when new GMO forms, there is a new copy of that desired gene of interest. Much more number of gene of interest have been produced in the growth phase in the fourth step. Now in this last step, all of these genes express themselves. They produce the desired proteins which were deficient and which were required to produce, for which we carried out the genetic engineering. Expression of genes means production of the proteins or products which were required.